Hello viewers, welcome to Mesology. In this video, I'll conduct an experiment to show you the process by which absorption of water occurs through the roots of a plant largely. Before I start with the experiment, I wish to explain certain terminologies associated with the process in order to make your understanding easier. So let's get started. Two glasses of equal size and volume. Let me mark them glass A and glass B. So here it goes as glass A marked in green. I hope it's visible to you. Glass A and then glass B. Glass B. So, two glasses side by side. They are of equal size as well as volume. Next, what will I do is, I will pour equal volume of water in each of them. So here, I will try to pour 150 ml of water in each. I am taking it from a measuring cylinder where I have 150 ml mark here. So I will pour 150 ml in each of the glasses. Here you have the marking cylinder here. In this, I will pour equal volume of water in each of them. So let me pour 150 ml in the first one. Little more. Little more. And now you are left with another 150. So it was a total of 300. Now another 150 is left. So I'm pouring this water in glass B. This is another 150 ml of water. Now both the glasses have equal volume of water. Next, what am I going to do? I will add certain solute to both the glasses. Now let me add a single scoopful of sugar in glass A. So single scoopful of sugar in glass A and stir it well to make it a homogeneous solution. Next, in glass B, I will pour 2 scoopful of sugar. I poured one, and then the second one also I will pour here into this. This is the second one. So I poured 2 scoopful of solute, that is sugar, into glass B. Now if I ask you, which one of these two is containing a dilute solution? Your obvious answer will be glass A, because both of them have equal volume of water. But glass A has only 1 spoonful of sugar dissolved in it. But glass B has 2 spoonful of sugar dissolved in the glass B. So naturally solute is more comparatively in glass B. Now if we have to say who is more concentrated amongst these two, the obvious answer is the solution B. On the other hand, if you have to say who has more water molecules per unit area, so it is the dilute solution who will have more water molecules per unit area. Why? Because less solute has been dissolved in it. And in that 150 ml of water, there is only 1 spoonful of sugar dissolved into it. But in glass B, there are 2 spoonful of sugar dissolved in that same 150 ml of water. So, speaking in terms of the water molecules per unit area, we have to conclude that glass A has more water molecules in its unit area as compared to glass B. So, we can say glass A contains a dilute solution. Similarly, glass B has 150 ml of water, but it has 2 spoonful of sugar dissolved in it. So glass B has less water molecules as compared to glass A per unit area. So glass B has water at its low concentration. Water is at its low concentration in glass B. I hope it's fine. So there are ways to speak. Either we can say a dilute solution, which goes for glass A. Let me mark it. I can write it. This is dilute solution. Dilute solution. So A is dilute compared to B, and B is concentrated solution. B is concentrated solution. B is a concentrated solution. So, in which of the two solutions water molecules are more per unit area? It is the dilute solution who will have higher concentration of water molecules but low concentration of the dissolved solute, whereas a concentrated solution will have more of solute particles and less of water molecules per unit area compared to the dilute solution. Perfect? To understand better the solution comparatively, let me pour a drop of food color into the more concentrated solution. So, the solution which is looking a little orangish or yellowish is the more concentrated solution, but it has water at its low concentration. Now we can correlate that a more concentrated solution has water at its low concentration and a dilute solution has water molecules at its high concentration. Understood? Next we progress towards a potato osmoscope to understand the process of osmosis. Now that you can see a vessel which is comparable to a petri dish, let me pour some water into it. So, I'm pouring some distilled water into the petri dish where water molecules are at its high concentration. Next, we have to take a potato and make a cube out of it. Or we can also cut in the shape of a cylinder or rectangular shape. Inside it, we will have to make a hole. So let me cut the potato. To make a potato osmoscope, I'm chopping off the side walls to make it easier to turn it into an osmoscope. I'm also peeling off the skin of the potato. Have a cylindrical shape of the potato. In this area, I have to, make a hole, or I have to make a cavity, a proper cavity in which I can pour a concentrated solution. But why pour concentrated solution? Because the cell sap in a plant generally contains concentrated solution because of photosynthesis that it performs. Let me fast forward this by making a proper hollowness inside this potato cube so that I can pour some concentrated solution which will actually resemble the condition of the cell sap inside a root hair cell. Let me make it better. Let me make this hollowness inside the potato cube a little deeper and then put it in the petri dish. So here it has almost become a nice hollow inside the potato. Now what will I do is I will put this potato cube in that water. I'm putting this potato cube in that water. And now the external environment, that is the water outside the potato cube, is distilled water. That is the most dilute form of water. So now it has water molecules. It's not a solution. Say it's pure water. So it has water molecules mostly concentrated. So water is at its high concentration in the petri dish. And inside this potato cube, where I have made a hollowness, what will I do is I will pour a solution with high concentration so as to create a difference between the two concentrations, that is inside the living cell and outside in the petri dish. Let me pour little amount of this concentrated solution here. Let me pour little more water into the petri dish. What we have to do is, we have to let it stand for some time. When we allow it to stand for some time, we will be able to make out the result after at least around 15 minutes. So let's see what happens. We will allow it to stand for 15 minutes. But before we move out of it, we have to mark the position of this solution inside the potato cube. So the level of that concentrated solution inside the potato cube needs to be marked. And after that duration of the experiment, we will recheck the level of water in the potato cube. So let us mark it. Marking it from inside where the water level is standing, or the level of the solution is standing inside the potato cube. This is perfectly the line that is touching the level of the more concentrated solution inside the potato cube. Whereas in the petri dish that we have kept the potato cube into, it has a low concentration of the solute, or we can say it is pure water in the petri dish. Either you use pure water or you can use a dilute solution where solute concentration is less. Now what will happen? Now let's check the level of the solution inside the potato cube. There was a pin inserted right at the level of the solution which touched inside the potato cube. Do you see the same? Or the pin which was inserted is now deep into the solution. So what has happened? Water molecules have moved inside into the burrow which has been dug into the potato, that is taken as a potato osmoscope. But why did the water move from outside into the potato cube? The net answer is water was at its high concentration in the petri dish. In this petri dish, water molecules were at its high concentration. And here inside the potato cube, it was a more concentrated solution. So, a more concentrated solution has less amount of water molecules per unit area. So, water was at its low concentration inside the hollow of the potato cube. We can compare this hollow of the potato cube to the root hair cell, because a root hair is unicellular, and inside the root hair cell the cytoplasm is with a big vacuole, which is indeed seen to draw water inside from the soil, and this is due to the difference in the concentration of water outside in the soil water as compared to the cell sap. So we can now explain it very clearly in two different ways. We can say osmosis is the movement of water molecules from its high concentration to its low concentration, crossing through a semi-permeable membrane. That means you have to have a living cell or a organ or any of these vegetables or fruits where you make a hollow and fill that with certain concentrated solution, making it resemble the condition prevailing inside a root hair cell. Now, this level of the water has gone much higher, and that is only because of a kind of osmosis which is called endoosmosis. Now, what is this endoosmosis? Endo is for whom? Endo is for inside the cell. So the word endo, which is used as a prefix, is indeed used for the entry of water inside a cell. And if the water would move out of the cell into the external environment, then it would be called as exoosmosis. Now, when can exoosmosis occur? When the external environment, which is shown here, the petri dish which is holding here distilled water, if this was replaced by a solution which was more concentrated with solute compared to what is filling inside this potato cube. If this solution is replaced by a more concentrated solution, then indeed water will flow out from the potato cube into the petri dish. That will be known as exoosmosis. But what do we call this medium to be, which is lying outside the cell in the external environment? We generally have special name for this solution. We can call it hypotonic solution when this is having less concentration of solute, then it will be hypotonic in nature. If it is replaced by a solution which has more concentration of solute, doing it for you to make you understand better. So if I'm filling it with a solution who will have more concentration of the solute into it, in that case, actually let me show it to you by adding. See, I have added quite a good amount of that solute. Now this solution in the petri dish is having even higher concentration of the solution compared to what is in the inside cavity of potato. Now, if this is kept for some 10 minutes or higher, water molecules will tend to move out from the hollow of the potato cube into the external environment, resulting in exoosmosis, and that will result in the lowering of the level of the solution inside the hollow of the potato cube, and the cells of the potato cube will shrink. It may also get plasmolized if it is not put into water immediately. Thank you for watching. If you liked my video, do not forget to give it a thumbs up. Also share it with your friends, and do not forget to subscribe to my channel. Thank you once again.